Hi everyone. I would like to introduce myself. I am V.Lakhmi, Assistant Professor, Department of Pharmacology, DRPI College of Pharmaceutical Sciences, Bengaluru. I am happy to present the project titled The Pegylation Epigenin Encapsulated Stealth Nanoliposome Promotes Superior Drug Release and Pharmacokinetic Profile, ensuring robust promise in hepatocellular carcinoma. The co-author, Mrs. Momita Dara, Assistant Professor, Nitya College of Pharmaceutical Sciences, Bangalore, under the guidance of Sir Professor Biswajit Mukherjee, Department of Pharmaceutical Technology, Jadavpur University, Kolkata. I would like to express my gratitude to Dr. Kusum Devi, Principal, Nitya College of Pharmaceutical Sciences, Bengaluru, and the organizing committee for giving me a beautiful platform, DRPI 2022. To begin with, the introductory part, hepatocellular carcinoma is currently positioned at the sixth rank in the cancer death worldwide. The reasons being the chemotherapeutic unresponsiveness are drug resistance, recurrences, therapeutic side effects, and the unpredictable drug accumulation in target tumors. With the development of nanotechnology, many lipid nanocarriers shows promising delivery for chemotherapeutics in cancer treatment. Yet, there were some challenges for meter dose accumulation versus clearance rate. In this project, we would like to highlight about the pegylated nanoliposomes that could expand the circulation time as well as the precise and controlled drug release in the tumor site. The bioflavonoids like epigenin, quercetin, curcumin have already gained enough interest in the cancer therapy for the nanotechnological approach in the cancer drug delivery. Here, the unexplored areas of the bioflavonoid epigenin, that is, pegylated stealth nanoliposome, were developed, characterized, and comparing it with plain epigenin nanoliposomes. Finally, the cytotoxic and apoptotic potentiality of optimized pegylated nanoliposomes were evaluated against the hepatocellular carcinoma. The objectives of the project were the development of the flavonoid-based epigenin, that is, the pegylated stealth nanoliposomes for the hepatocellular carcinoma treatment, optimization of the particles like zeta potential, size, surface morphology, and stability for the prepared nanoliposomes, ensure sustained and metered in vitro drug release patterns with respective kinetic models, and the in vivo pharmacokinetic assay for checking bioavailability in animal models while observing tumorogenic uptake both in cellular and tissue sites. Coming to the method of preparation of the formulation, the development of the nanoliposomes is done by thin film hydration technique. The formulation ingredient compatibility is estimated by FDIR technique. This is the slide that clearly shows the optimization of the formulation done. The particle size distribution, the zeta potential, and the percentage drug loading efficiency were determined. Three different formulations with the name F1, F2, F3 were prepared with the composition of the cholesterol, soya lecithin, and DSPE PEG 2000. For the formulation 1, it is found to be having the average particle size of about 45 nanometer and the zeta potential ranging -4.22 and the percentage of loading efficacy is found to be 92.10%. For the formulation F2, the particle size is in the range of 95 nanometer with the zeta potential of around -55.9 and the percentage of the loading efficacy was found to be 81.57. Whereas for the formulation F3, the average particle size is found to be 143 nanometer with the zeta potential of -53.5 and the percentage of the loading efficiency is found to be 76.31. The graph that is presented below in these slides as F1, F2, F3 are nothing but the images of DLF particle size analyzer. From this data, it is very clearly understood that the formulation F1 with the average particle size of 45 nanometer and the zeta potential value of around minus 4.22 with the percentage of loading efficiency around 92.10 is found to be best suited for conducting the in vivo and the in vitro studies. This is the slide that clearly explains about the morphology of the pegylated nanoliposomes that was formulated. This morphology indicates the uniform distribution of spherically intact and bilaminar stable formulation that has superior metered dose drug release pattern, which is analyzed through FESM, cryotem, and AFM techniques. In this slide, the in vitro drug release is clearly shown. After plotting the drug release data in different in vitro kinetic models such as zero order, first order, Higuchi, Coase-Meyer-Pappas, Dixon-Crowell, we observed that here the drug release pattern is best suited with the Higuchi kinetic model with the retention value of around 0.9811. Here, in this project, the pegylated nanoliposomes is compared with the plain nano epigenetic liposomes, and this study is conducted at the different pH values of around pH 7.4 and pH 5 with 1% beta cyclodextrin in phosphate buffer. For the in vivo pharmacokinetics, it is very clear that the data also approach much extended area under the curve and MRT values for the pegylated epigenetic nanoliposomes is much more than when compared to the plain epigenetic nanoliposomes. The conclusion of our project is after developing the pegylated nanoliposomes with the size less than 40 nanometer in range and coated with PSPE PEG 2000, we concluded the in vitro drug release for the pegylated nanoliposomes was superior than the normal liposomes. It showed desired prolonged sustained and metered dose drug release. The in vivo superior bioavailability, almost doubling the AUC and MRT indicated the extended systemic circulation advantages for PEG NLCs over normal nanoliposomes. These characteristics of PEG nanoliposomes probably allowed for tumor uptake of epigenin-loaded pegylated nanoliposomes and their robust promise on curing carcinomas, where in future we specifically will conduct the studies for targeting the neoplastic hepatocytic cells. Thank you.